Under research has mentioned a Paraquat induces cellular aging and leads to cognitive diseases. Well, are there any other substance may cause the similar effects?
Yeah, so Paraquat, as I mentioned, is this pesticide, which frankly has been banned in a lot of European countries, but is still in use in the US. There are these epidemiological studies that show that there's an increased risk for development of PD in people that have been exposed, as well as preclinical models like worms, mice, non-human primates.
Well, it
And
seems that Paraquat is not forbidden in China too.
yeah, or Brazil, actually. It's very interesting. In fact, there have been some lawsuits brought against some of the manufacturers of major manufacturers of pesticides involving some of these farm workers that that developed. Yeah. So as you alluded to a few years back, and this is one of the first studies to look at a model of Parkinson's disease, we found that so it's known that if you give Paraquat at a certain dosage to mice, they develop Parkinson's disease. You get loss of these midbrain dopaminergic neurons, you get increase of alpha-synuclein, the mice show motor dysfunction, and we found that if we genetically eliminated senescent cells, we could prevent development of Parkinson's disease in that model. And that paper was published in 2008, early 2018 in Cell Reports. And then this field has kind of exploded since then. On the heels of that, there were a couple papers which came out looking at different preclinical mouse models of frontal temporal lobe dementia, which is related to Alzheimer's disease. So there's been a huge interest in cytolytics. In terms of like how does Paraquat work, it's known to be a redox cycler. So in other words, it produces oxidative stress, which can kill vulnerable neurons. And the levels of oxidative stress are known to be increased in brain regions in PD patients. And oxidative stress can induce senescence. So that may explain why there's an increase in levels of senescent cells, both in postmortem Parkinson's brain tissues in the substantia nigra and when you give mice Paraquat as a model of PD. There are a number of other substances that are known to emulate features of PD. I mentioned rotenone. Rotenone is something that has been used to call fish populations within lakes. It's actually an inhibitor it's a mitochondrial inhibitor, so it actually acts to prevent this organelle within the cell from producing energies the cell need to function. And if you give rotenone at a particular dosage regime to mice and rats, they show signs of Parkinson's disease. MPTP, which was something that was discovered in the late 70s, really by dint of the fact that there were drug addicts in the Bay Area in San Francisco that were trying to make, in essence, a heroin analog. So they ended up making this compound MPTP that when they injected it, and it's very sad because these were young men, and they ended up with kind of an acute Parkinsonism. And people again realized that this was emulating what you see in PD, and it was used for several years as a model in mice Parkinson's.
That you have mentioned the mice, they will grow up symptoms or implications of cognitive diseases. Did the researchers raise them with any other drugs that may lighten their symptoms or treat the diseases?
Yeah, so a lot of before we really understood some of the genetics behind familial Parkinson's disease, the models that were used were really these, what I would term neurotoxin models, where people would, for example, give a mouse MPTB, and the mice would sort of have this acute development of phenotypes, neuropathology, behavioral, motor effects that you see in Parkinson's patients. And these were actually used to test different therapeutics. The downside to those neurotoxin models is they tend to be, unlike what happens in Parkinson's disease, which is development of the symptoms and the pathology in an age-related manner, and also kind of slowly. These animals would develop Parkinson's very rapidly. Even in young animals you could produce. So like for myself, it was somewhat problematic in that you're not really then looking at the contribution of aging. So I will say for these original senescent studies, we did use Paraquat. In a way, it was because it is rapid. The pathology is due to production of oxidative stress. Oxidative stress is known to produce senescence. Since that time, a colleague of ours, Juan Kim, at University of Delaware, has looked at senescence in the context of more chronic models, like genetic mouse models that are expressing human forms of mutated alpha-synuclein. And they've also, in those models, seen that this increase, you have an increased presence of senescent cells. I think we're still trying to sort out in the AD and PD research communities what cells undergo senescence in these diseases, and does it different from, in a sense, although we talk about Alzheimer's and Parkinson's disease, these are probably syndromes. Different people present in different ways. And again, it's very complicated because it's a mix of not only increased risk with aging, but also as individuals, we all have a particular genetic makeup. We all have particular lifelong environmental exposures, and that can all, in a kind of complicated way, contribute to whether someone's going to end up presenting with these diseases. So I can say in some of both the AD and PD models, depending on the model, you see different cell types undergoing senescence. So I think a lot of times biology, we start out, especially for people like myself that are sort of molecular biologists, we start out with very simple hypotheses, and biology is complicated, so things start getting a little more complicated as we look at them a little bit more. But a very exciting advent, because so much emphasis had been put on plaques, A-beta plaques, and neurofibrillary tangles that contain this protein tau. Those were kind of the two neuropathological hallmarks that are associated with Alzheimer's disease. And a lot of the research over the last 20 years was towards making antibodies that would eliminate those pathologies. And until recently, clinical trials using that type of immunotherapy wasn't very successful. There have now, over the course of last year, been a couple of trials that seem to suggest that immunotherapies against A-beta plaques actually seem to slow cognitive loss. The flip side to that is that because those immunotherapies are bulk and they don't just target the plaques, they can also target things like the vasculature in the brain. And so a serious side effect of use of these type of immunotherapies is it can cause brain bleeding. And that can lead to death in a subset of the patients, which is another reason why I think it's, very important that we think about how to target these medications to the pathology. So yeah.